So, hello everyone. We are on episode number 4 of Clinical Research Talk series, and the topic of today's episode is clinical trial design and phase analysis. So, in this particular session, we are going to talk about different clinical trial design right from phase 0 to phase 4, and also what are the statistical analysis methods used from one phase to another, because you have to understand that when we get approval for phase 1, phase 2, phase 3, so there are changes in statistical analysis, and these particular analysis is what leads to approval of the medication. So without further ado, let's start this video. So I would like to inform that this particular series is in collaboration with Aditi, so who runs the Alpha to Omega channel, and also Clinical Aim Research and Clinical Research X360s. These are YouTube channel. We are also on Spotify, as you can see your Clinical Research Talk series, and you can even listen to our sessions in terms of and save some time. So let's start. So let's understand the different phases of clinical trial. So clinical trial are in generally considered to be conducted in 3 phases, from phase 1 to phase 2 to phase 3, and then the approval. So before submission of phase 1 to test in humans, there are preclinical studies which are done in animals. Okay, so that phase is called as preclinical phase. After that, it moves to phase 1, where the number of participants change from 20, and also the number of indication or focus area changes. So in phase 1, we look at safety, where phase 2, we look at safety and efficacy, and so on and so forth, until we have the FDA approval, and that particular medication then can be given to the masses. So this is in general about various clinical trial phases. So let's look at each phase in detail and also get to know about the statistical analysis of each phase. So let's start from phase 0. So phase 0 is the phase called as first in man study, because here before this phase you have done preclinical trial which was done on animals, but in this particular phase you are introducing the drug for the first time in human beings. And although the genome of some animals and humans can be 99% similar, but there can be some dissimilarities which could lead to adverse effects. So that is why phase 0 is critical. Here the number of participant is only 20 to 30 healthy participant, because here we are not focusing on the effectiveness or efficacy of the drug, but here we are giving a very low drug, less than 1%, just to check the clinical effect of it and if there are any adverse effects. That is the focus. And the aim of this particular phase is to test between chemical analogs and to see if there are any intended clinical effects. So that is phase 0 for you. Now let us understand the statistical analysis of this phase.
Yes, so as mentioned in phase 0, we are not necessarily testing any sort of clinical effect, although we may. It depends from trial to trial. But we are testing, we want to see how the trial is how the medication affects humans. So what we observe is adverse effects, and also in inferiority or superiority or non-inferiority between chemical analogs if applicable. So here what we do is that we are getting the summary of approximately 20 patients in different kind of cohorts, and assuming that we have, say, a couple of cohorts between chemical analogs, we do the statistical summary of each of the patient. And it's very important to consider the statistical summary of an individual cohort separately, because that's what we want to know at this point of time or at this early phase. And then here we choose the statistical analysis method according to the endpoint. So in the last series, in the last episode, I had mentioned about endpoints and how endpoints are different kind of endpoints can determine the statistical method. So for example, if we have a continuous endpoint or continuous data, then we would go for a different kind of endpoint. We would go for a different kind of statistical significance. Maybe we will try a null or alternative hypothesis endpoint. Whereas if we have continuous data, we might go for t-tests or other sort of statistical tests, which is according to the endpoint that we want. And here we do compartmental analysis to understand the pharmacokinetic parameters. So this will be in a very generalized pharmacokinetic parameter, since it's not done in patients, because here we choose healthy subjects. So here we will understand the pharmacokinetic parameters in healthy human subjects, and understand how the drug pharmacokinetic is. And we could also do covariate or multivariate analysis to understand the effect of covariates on the given dose or the drug. Now, what is covariate or multivariate analysis? So, our covariates can be considered as input variables or values that do not have direct effect on the dose, on the drug or the effect. For example, uh, weight or height or sex or renal efficacy, this could be considered as a covariate, because though it is not the input value, because for example, something that would be directly affecting the performance of the drug could be the dose or could be the chemical composition of the drug. But this is not the direct input value, but more like an indirect input value, which could affect the drug performance. So these are what we call covariates, and there could be multiple covariates. So hence we need to do multivariate analysis to come to a conclusion and understand the effect of covariates. So maybe we could do individual covariate analysis, such as understanding the weight separately or understanding the effect of the height separately, or it could be combined. So here we also, as discussed previously, we can build a basic pharmacokinetic pharmacodynamic model, which can be later escalated in specific patients. But here we do a very generalized pharmacokinetic pharmacodynamic model to understand the dose and covariate effect. So I mean, we can build a basic structural pharmacokinetic model, which is whether to understand whether it's a one compartmental, two compartmental, or compartmental multiple compartmental drug. And I mean, if the pharmacokinetics are like that, and also understand how different kind of covariates can affect the pharmacokinetics. And based on this finalized uh, pharmacokinetic model, which will be further validated, we can build a pharmacodynamic model to understand the pharmacodynamics, like whether it's a direct pharmacokinetic model or an indirect pharmacokinetic model, or is it a delayed pharmacodynamic model. And here we kind of establish a very basic pharmacokinetic pharmacodynamic model in healthy patients, and also we can plot outcomes against one another, or do a regression based analysis. Now, what kind of outcomes can we plot against each other? So maybe we can, a very basic outcome to plot against each other would be the dose and time effect, or maybe we can even plot, say, the weight against the dose effect, or other things that we think are important to see. And the reason that we plot outcomes against each other is that we understand what kind of a relationship they have. Like maybe they have like a linear relationship, whereas a simple linear relationship could be that in patients with higher weight, maybe the concentration would be higher due to various reasons, or a patient with lower weight could have other kind of relationships. um, yeah, so you could have a direct and indirect effect with plotting outcomes within each other and understand whether they have a linear relationship or not. And here at this point, you could also do, if applicable, regression analysis to predict different kind of outcomes. Though many a times you don't do regression analysis at this phase because you don't have enough of data. Now, what is regression analysis? So regression analysis is basically to understand, when you plot against an xy value, to understand the different kind of, to understand the mean and understand the outliers of that value. But as mentioned before, many a times you don't do regression analysis at this phase because it doesn't have enough of data and conclusion. Yeah.
So now let us move on to phase one design. So we have talked about phase zero. In phase one, what happens is we are evaluating three typical items here. First is the safety part of view. Second is the efficacy, and third are the common side effects with dose range. The thing is, in phase one, we focus on a limited number of participants, again 10 to 15. But here, what we are focusing on a single dose. We are also testing the dose range, what we are planning to give in this particular clinical trial. We are deciding whether the range should be X or Y, and we test this range in kind of ascending model, where we go from lower to higher just to avoid the adverse event, and also give us an idea that what are the immunological effects shown by the subject at a particular dose. So that is the focus of it, and the main aim of it is obviously the dose selection and what is the maximum tolerated dose, which that means the dose at which we see the maximum immunological reaction or maximum adverse event. So here in phase one, the focus is to selection of the dose to see the adverse event and what are the final dose concentrations of it. So now let us see what is the statistical analysis part of phase one study.
Yeah. So as mentioned before, here we want to find out, like, different kind. We want to explore. We are still at the exploratory phase, so we want to look at different outcomes, different patients, and also different outcomes in the single ascending dose study as well as multiple ascending dose study. And in usual first we have single dose ascending study, and on based on the statistics of that, we go to a multiple dose ascending study. Although there are times that they happen at simultaneously in different cohorts. Therefore, it's very important to have, again, we have statistical summary of different kind of patients and their outcomes, because we need to see what kind of outcomes they have, like depending on what kind of drug we're selecting. And we need to have a statistical summary of that, and at all points statistical summaries are really important. And we need to look at whether there's a difference between the statistical summary at phase 0 and phase 1. And now we look at visual representation of individual variable of input variables versus outcomes, where we see if different input variables have an effect on the outcomes, whatever they could be. And also here we understand the absorption phase and elimination phase of the pharmacokinetics, like at what phase at what time and at what concentration does the drug absorb, and also at what time and concentration does it eliminate. And these kind of this information also kind of helps us in the calculation of individual pharmacokinetic parameters such as Cmax, Tmax, bioavailability, elimination constant, half-life, etc. Now here, since we have an idea of the pharmacokinetic parameters in a group, we also can calculate individual pharmacokinetic parameters, and we statistically summarize them to understand more. And now per dose, because since we have single ascending dose studies as well as multiple ascending dose studies, we will also take the pharmacokinetic parameters and calculate them. And then we will take a statistical summary of those per dose, because through that we can actually understand more about the dose, understand more about the clinically significant dose, and also the dose that we do not want, because in case they're causing adverse effects. So this also kind of gives us final statistical analysis outcomes per dose, so that kind helps us in our dose selection. But and hence we are exploring different outcomes in different doses. And as mentioned in the previous phase, we also do covariate analysis here, because since we're doing a single ascending dose study and a multiple ascending dose study, we need to understand different variables that can influence the dose. And pharmacodynamic effect is analyzed finally, and we develop the pharmacodynamic model and see if there are different changes between the previous pharmacodynamic model, or do we follow the same pharmacodynamic model. Yeah.
So now we move on from phase 1 to phase 2. So in phase 2, it is also called as therapeutic exploratory phase, where the efficacy is focused on while maintaining the efficacy at in different number of subjects. Here the number of volunteers or even the volunteers in some medical condition go from 100 to 300. And these volunteers in case of vaccine can be healthy, or in case of other clinical trial, for example diabetic clinical trial, they can be diabetic subjects. Here a particular medical condition is targeted, and your multiple doses are tested against the placebo. So why do we include placebo in clinical trial? It is because to understand whether the given medication is having its effect or efficacy based on its chemical formulation, or is any other factor such as the mindset of the subject, or the belief that a particular medication shall cure a particular disease, is that affecting the effectiveness of medication. That is why a placebo is given. So a placebo against the actual drug is tested, and aim of this particular phase is to explore the clinical effects against the targeted disease. So how is this particular medication reducing the adverse event, or reducing the progression of that particular disease, and also understand what are the adverse effects, because you could be curing one thing and another thing could show you in terms of adverse event. So that is also a main focus area of phase 2 design. Now let's see what are the statistical analysis of phase 2 study.
Yes, so now our data is expected to change completely, because we have patients in this cohort, we have patients of that particular disease, and also understand it that if a patient has one disease, they could have other compromised factors. For example, maybe they could be cancer patients, but they have compromised renal function and other problems. So we expect that our data will be nothing like the data of healthy subjects. So we have to repeat the statistical summary of different parameters. We have to do the statistical summary of uh, the different kind of parameters with respect to the dose, as also with respect to their own covariates. So covariates such as uh, the weight, height, and other
Comorbidities.
variables of patients, which will be very different from healthy subjects, as mentioned. So we do the pharmacokinetic analysis again, and here we will focus on the parameters that define like the pharmacokinetics, like absorption, metabolism, excretion, and distribution. And this will be here we will understand the pharmacokinetic parameters in patients. And here the we define the pharmacodynamic effect. We put a lot of focus on the pharmacodynamic effect. So either we will focus on certain biomarkers, or we will focus on concrete endpoints. So we assume we assume that the pharmacodynamic model is made at this point. We understand the model, And here we will focus more on the biomarkers and concrete endpoints. And the statistical analysis of this phase is very important because here we want to select a final dose for the phase 3 study, and we want to finalize clinical endpoint, and this will lead to dose selection in patients. Now the statistical dose selection is important because we need to make sure that it's analyzed in a way that they have minimum side effects, but also the final pharmacodynamic effect should be uh, at a clinical level, that that they don't that they should have more than a minimum effect. So these are very important in terms of the statistical phase 2 analysis. So here you could also look at different kind of aspects, like maybe you could look at a T test or a Z test, or even determine the statistical significance at this point. And this would vary according to what kind of data you have particularly. And this is one of the most statistically important phases because we are determining this for patients, and we from the next phase onwards we are going to go to a more heterogeneous group of patients. And this statistical the final statistical outcome will end the exploratory phase, and this will be the confirmation, like the statistical confirmation of what we already know from the previous trials. Yeah.
So now coming to phase 3 design, now we have, as we have seen in phase 2, our dose is decided now. Now what we are focusing on the exploratory effects of the effectiveness of this particular drug. So what we want to do here is, now we have a fixed dose and we want to test that particular fixed dose in a larger population. This larger population, as Aditi correctly said, it is going to be heterogeneous population, not only selected to one particular area or limited number of patients. So number of patients increased tremendously from 1,000 to 3,000, and this is just a standardized number, 1,000 to 3,000. It is more than 3,000. Okay, it can be 10,000, 15,000 also. And these particular volunteers are targeted for a very specific medical condition for which this particular medication or the molecule is being marketed. Here also you can test multiple ascending dose, but mostly the dose is fixed in phase 2, and we focus on the adverse effects and the efficacy part of it. So aim of this particular phase 3 would be the confirmation of clinical effects, okay, against the targeted disease, and also understand that how this drug performs in terms of efficacy in larger target populations. Because this is the last opportunity for the study trial investigators and the sponsor to test their medication, because after phase 3 we are going to submit all the clinical data to the regulatory authority, and post which they will analyze the data and provide marketing approval. So this is one of the biggest phases of clinical trial. Now let us see what are the statistical consideration of phase 3.
Okay, so at this point, we have a very heterogeneous group of patients, and we also will have a lot of heterogeneous data because here we also prefer like 1,000 to 3,000 patients, which is a lot of data, as well as it could be a multi-centric trial in different countries. So we could get different results from different, say, different races or different kind of patient groups that we're looking at. So again, here we can do a statistical summary of patient demographics. We can, which we have to look at the dose that we are doing it. We have to do it in the particular cohort. We have to be very particular about that. And adverse effects have to be taken individually and together. Now, the importance of taking of statistically summarizing everything individually is that we understand the effect of the dose in that particular group, and that could be very important from a local regulatory perspective. And also we understand if there are certain kind of varieties of adverse effects in the particular group. Also it is important to take the statistical summary together. So I mean in two different data sets, so together can give a more global perspective of the drug. So yeah, we do the statistical summary together and individually. And also here we can have an application of machine learning, which could identify different kind of outcomes, as well as it could predict different kind of outcomes, because we have enough of data here and we have enough of heterogeneous data here. In the previous phase we had smaller data sets and maybe not so heterogeneous data sets, so machine learning was not preferable at that phase. At this phase, we can use machine learning for a number of things, as mentioned before in our previous episode. Uh, Here also we do PKPD models, because this is important at each phase. It's important to do it because here we are getting a heterogeneous population and we need to understand their particular covariates, and we need to understand whether we make decisions based on those covariates. And we hope that at this level, the PKPD model is is standard, that we don't have to make a PKPD model, but more like we follow a PKPD model. And through that, we can understand their own, how their individual covariates affect the PKPD model. And this can give us a lot of information, such as the final dose and the final outcomes, and a lot more information, because PKPD model can vary from very simple to very complex, so that can give you a lot of information at each point. And we do statistical tests separately when we like t-test, ANOVA, hypothesis testing. And again, as mentioned before, this depends on the endpoint. So this usually gives us the final statistical decisions, whether there is an inferiority on an or a non-inferiority, and the final statistical analysis. And these are the statistical endpoints that we will submit to the regulatory authority in order to get approval for marketing. Yeah.
Yeah. So once you get the approval for marketing, clinical trial does not stop there. There is a phase 4, which is called as post-marketing surveillance. So now, till this point of time, till phase 3 study, we were giving a particular medication for certain medical condition. For example, we developed a drug for diabetes. But when that drug is prescribed to a patient in the open market, and when it is used by everyone in the world, you cannot restrict medical conditions, because now here there will be a subject who has diabetes along with anemia, or who has other kind of comorbidities. So here the number of drug interaction, the number of interaction with other kind of medical comorbidities increases, as well as the number of subjects. Now in phase 3 study there were 3000 subjects, but when the drugs get marketed and when you can use it freely, or when the doctors can prescribe that particular drug, then the number of subjects increases, and it creates a lot of data. So after marketing of the drug, so for 20 years, post-marketing surveillance is done, which is generally referred to as phase 4 study. Here, the number of participant is not limited. It can be all the subjects who are using this particular medication. And when it comes to dose, here there is a variation, because in phase 3, during regulatory approval, we recommended that this particular medication is showing efficacy at this particular dose. But based on the comorbidities, health condition, gender, what kind of effects that the particular medication has, the dose is variable in from one subject to another. Therefore, dose can also create various uh, effects on the subjects. And why we do this phase 4 trial is to understand that what are the drug interaction, or what are the adverse event that the manufacturer or clinical trial investigator missed during the clinical trial in phase 3. So to understand the effect of the drug, to understand the adverse event that are being caused after marketing, So that is where phase 4 comes in. And please remember that phase 4 is not only in terms of surveillance, but it is also advised by the governments over the country, because it is not just approval is not just granted for the entire lifetime. Approval is granted based on that it does not create any negative adverse event. If there are cases reported of negative adverse event, then that particular drug can be banned or stopped, and again the clinical trial can be recommended. So the sponsor is also very active, the pharmaceutical company, in phase 4 design, and they focus on the clinical effects after the approval and what are the side effects, and they constantly monitor during phase 4. So the number of data generated during phase 4 is tremendous. And now let's see what happens in phase 4 data analysis.
Yeah. So phase 4 analysis is when we get into like a phase called pharmacovigilance analytics. So this is basically very complex and also a much larger data. And here one uses advanced analytical techniques with the purpose of examining a lot of data as well as a variety of data sets. And it contains safety information, and you can also use machine learning to uncover hidden patterns, unknown correlations, trends, and patient preferences, and several other useful information that can help, say, organizations like pharmaceutical companies or health economic companies or contract research organization or regulatory organization to make more informed decisions. So what is unique here is that we have big and heterogeneous data analysis. So here one can actually use standardized statistical analysis, I think, and one can also be using machine learning. So what kind of machine learning can be used here or would be very effective? So here unsupervised machine learning would be really effective. As mentioned in the previous video, unsupervised machine learning is when data is not labeled in any way. So you're not getting a data set with this thing that you have to predict. You're using a lot of heterogeneous data around the world with the marketing drug and the outcomes. So here the most common use of unsupervised machine learning is to do clustering analysis. So in the sense that if there is a side effect that is reported, then it is clustered together, it is sorted together with the likelihood, and there this sort of clustering is done, and then from there the clustering could be analyzed to understand whether, I mean, there the clusters can be statistically analyzed to understand whether there are more side effects occurring in a certain population, or other kind of trends that can be statistically analyzed. And also to give clustering can also, unsupervised machine learning can also help understand different kind of relationships between data. And this is done by reducing the dimensions of data to a smaller dimension to understand if there are certain kind of relationships between data. But it is not uncommon to also use supervised learning here, because supervised learning is can be gathered from a labeled data set. So this can be done when there is a particular data set or particular amount of structured data about certain kind of side effects or other effects that have occurred. From here, one can predict if there is a certain kind of trend. So here, supervised learning can be used. And one other very important use of unsupervised learning is if if a drug has a certain effect and if that needs to be or can be repurposed, because unsupervised learning or deep learning, it is capable of understanding certain unlabeled data and understanding if there is something other than the intended effect, and whether that drug can be used for another purpose. So this is a lot of useful data that is coming out, and that can be structured. Now, here one can also do outcomes to adverse event ratio analysis. So this is the same as understanding that why a certain adverse effect has occurred, where has it occurred, what are the outcomes, and what is the ratio in a certain population. And that can be taken as individually, uh, as individuals data as well. Now, trend analysis, as I mentioned, that maybe there was a certain adverse effect that was observed in certain populations. So this can be done through trend analysis, and yeah, so it can also, trend analysis can also help make local regulatory decisions or local decisions regarding the drug, and as well as can be helped in repurposing the drug. So now, as I've already talked about clustering and machine learning based analytics, and its wide applications, I would also like to talk about signal detection. So signal detection is basically when certain adverse effects are reported, and the agencies that are responsible for drug safety detect these. So this can either be done manually, either they can be picked out, and they can be in a different data set, which will be further analyzed in the future, or it can be done automated. So how is automated signal detection done? Well, it is done. This is also done in an unsupervised way, because uh, the common side effects can be labeled, and they can match against a database of known side effects, or there are other ways of also labeling the side effects or the signals. Now, there are other ways like natural language processing, or text automation or text mining, that can also automate these processes. So there are a variety of ways of automating signal detection. However, a combination of automated and manual signal detection is very important, because oftentimes automated signal detection can fail. For example, if we have a certain side effect and it's labeled in a certain way, uh, the I mean, understand that the whole world does not speak English. The whole world does not have the same proficiency that is there, that would be there in a labeled data. So maybe they could make a typo, or they could not report it in that way, or they could report it in their own language, so that would not match to the database that, or the label data that we want. So hence we would get a lot of confused data. So hence it is very important for the drug safety to have manual manual detection, and in the future to improve signal detection, to understand that, and include like say other language in automated signal detection, or make a software sophisticated enough to understand that there could be certain common typos. So yes, so this is the how in an overview of how the post marketing data is analyzed. Yeah.
Yeah, and also I would like to add that I mean, there are agencies who look and focus typically on pharmacovigilance data and signal detection. So globally, it is the WHO Uppsala Monitoring Center who takes care of the global pharmacovigilance signal detection. Also, this particular agency lays down global guidelines for pharmacovigilance programs and how analysis should be used. So this is the premier agency for pharmacovigilance, and also when it comes to country, they have their own individual pharmacovigilance program, such as India has the pharmacovigilance program of India. Okay, so PI also clearly explain what are the pharmacovigilance guidelines that a particular regulator must, that a particular sponsor must follow, and how we must monitor the signal detection when it comes to post-marketing data analysis. So this is about phase 4 post-marketing data analysis. Now let's talk about what are the next things we are going to see in this particular series. So when it comes to the future, we are all excited and we want to know that what are the upcoming exciting aspects of clinical research. So in future series, we are going to talk about the decentralized clinical trial, because as we know, that clinical trial are conducted globally and multi-centered, but this is a new concept called as decentralized clinical trial. So so we are going to see what are the aspects of it and how this can shape the future of clinical trial. Also now, since the advent of AI and machine learning, we have seen the in silico clinical trial. So how machine learning or how AI can assist in silico trials. So how these can compute a large number of data and how that can reduce the cost of medication directly. So that could be another exciting option for the future. And we are also going to shed light on the risk-based monitoring. So RBM is a very hot topic currently. So we are going to also look at RBM and its aspect. So what do you think, Aditi, about it?
Yeah, I'm really excited for this because the world is changing, clinical trials are changing. Finally, the recognition of them it has come to light, and I'm really, as usual, in quite excited to talk about the in silico aspect because I've dealt with some of these softwares myself, and uh, to talk about risk-based monitoring and other aspects. So I'm really excited, and we're going to talk about different kind of trials. We're going to talk about trials that have been done. So we have something really exciting. We have something that we are really going to learn a lot while making these videos, because it's not just that we put information out there. We also learn while making these videos. So
Of
yeah,
course.
and this is the bibliography and the references without which we would not have been able to put information out there. And I mean, as usual, that we're very thankful that people put information out there, and we hope to contribute to the community of information. So thank you for tuning in, and we are really looking forward to our next video on the future of clinical trials next week.
So stay tuned and thank you all of you.